In this video, I'm going to take just sort of a brief look at the biochemistry of the Delta variant of COVID-19. And so I got this figure here from this article right here called How Dangerous is the Delta Variant, which is oftentimes called B.1.617.2. This is on the American Society for Microbiology website, and I'll have it linked to in the description down below. But what this is showing is the spike protein and different mutations within the spike protein. So what we have up here at the top is the amino acid positions within the spike protein, and then we see this part right here is the receptor binding domain. That's the part of the spike protein that binds to the human ACE2. Then we have the furin cleavage site, which is the part of the protein that gets cleaved by enzymes in human cells, well, on the surface of human cells, so that it allows entry of the virus into the human cell. So what we have here are the mutations for the alpha, beta, gamma, and delta viruses. Alpha, I believe, is that UK version, which I did a video on a while back. But we're going to be talking about this Delta variant here. So the two mutations that seem to come up a lot in this Delta variant is this P681R, this proline at the 681st position mutated into an arginine, and then this leucine at the 452nd position mutated into an arginine. And this one actually occurs here in the receptor binding domain. This one occurs near the furin cleavage site. One other thing to notice too is so the first thing is this mutation right here, which occurred very early on in the lifetime of SARS-CoV-2 and has stayed with the virus throughout all of these different variants here. But then also this one, so this asparagine at the 501st position mutated to tyrosine, which was sort of the mutation of interest in this alpha version. It occurs here in the receptor binding domain, is now no longer present in the delta variant, which is kind of interesting. There are some deletions that occur down here near the N-terminal side, and so that seems to, if I remember correctly, have to do with the binding of certain types of immunoglobulins onto the spike protein. So these deletions sort of neutralize that those antibodies. But anyway, I wanted to look at a few papers here. And so this one, and like I said, I'll leave links to all these in the description down below. So this is in A up here is looking at, so this is looking at the Delta variant here. And it's looking at infection of different cell types. So the Viro E6 with this TMPRSS2, which is an enzyme that can cleave the spike protein. So that's one of those things that cleaves here at this furin cleavage site. And so it's looking at the infectivity in hours after infection of the cells here. And it's looking at RNA using quantitative PCR, quantitative polymerase chain reaction. And so we see what we have here, this B.1.1, I guess is sort of the OG virus. That's the one that came up. That's the one that occurred even before this D614G, this aspartate to glycine mutation here. So that's like I said, kind of the OG one, I guess you could say. And it's comparing the amount of virus present in the cells for that one, and then in orange the Delta variant. And so it infects these cells at more or less the same rate within the first 3 days. Down here in C is actually looking at the amount that is there after several days of after post-infection, and so we can see that decreases down here. In B here is something interesting. It's something I had not actually encountered till I was looking into this, and this is this Syncytia here. So there is this paper that I found, and I'll leave a link to this in the description down below, about the Syncytia formation by SARS-CoV-2 infected cells. And essentially what that is, oops, it's my references page. What that is we have our cell, so we have a cell here, and this one is infected by the virus, and when it's infected by the virus, it will actually start expressing spike proteins on the surface of the cell, and then you will have another cell, and it is not necessarily infected. In here I will put nuclei in here because that will be important here in a second. So these are the nuclei of the cell. But what this cell, whether it's infected or not, has the ACE2 on it. And so this cell, this human cell with the spike protein, can actually infect this other cell. And then what happens is we end up with these cells merging so that it's sort of like this half one cell, half the other, and it will have two different nuclei in it. And then this can happen with more and more cells, so that you end up with cells that, are these big sort of hideous things with a bunch of nuclei in it. And these are human cells that have essentially infected other human cells using the viral spike protein. And so that is what this Syncytia is here. And it's kind of hard to see here, but you can actually see this in these two sample images up here. So we see these big Syncytia, which are these sort of dark globs on here. Actually, let's see if I can zoom in a little bit. So we can see there are cells in here, and then these sort of darker spots are the Syncytia. Then with the Delta variant, we actually have more of these Syncytia. And this here is the measure of the floating Syncytia. And so we see uninfected is at 0, which is where we would hope it would be. With the OG virus, we see more Syncytia. And then with the Delta variant, we actually get a lot of these Syncytia on here. Down here is actually looking at the body weight of hamsters that have been infected, well, either uninfected, infected with the B.1.1, or infected with the Delta variant. And we see, so body weight being sort of a measure of how healthy the animal model is. So we see the body weight goes up with the uninfected version, and then with the infected, either with the B.1.1 or with the Delta variant, that the body weight goes down, which is an indication that the hamsters that they used were made sick by this. And so if we move down here, so this is actually looking at just this P681R. So remember, that is this mutation right here. So it's looking at just the P681R. So it does not have all of these other mutations in the Delta variant, just this P681R. So they're isolating that one. And so then they look again at the hours after infection using the quantitative PCR, and they see, well, they're actually comparing it to this D614G, which, remember, is this one here that I said was now ubiquitous in pretty much all variants of COVID-19. So they're comparing it to that, and we see that the infectivity of these cells based on how much viral genome is inside the cells in 24, 48, and 72 hours after infection. Once again, we see that we get more of these syncytia here when we add this P681R, this proline to arginine mutation onto here. And so kind of moving down here, what we see in E and F. So E is looking at how much of the virus is able to get into the cell in hours after the virus was added to the cells. And it's looking at it with minus this enzyme and plus this enzyme, which is one of the enzymes that actually cleaves the spike protein. And so this is showing that, once again, that there's more infectivity in orange here with when we add that P681R mutation that we find in the Delta variant. Then an F over here is looking at the speed of infection. And so we see that right at infection that this P681R is so it says fusion velocity, so how fast it is fusing with the human cells. And I mean, it ends up going down below the D614G only one here, but that, I don't know, I guess it kind of makes sense if you think all these viruses are getting into the cell super fast. It's going to slow down as the concentration of viruses outside the cell decreases. But anyway, once again, what we're seeing here, the takeaway message is that this P681R mutation is increasing the infectivity of the virus, and that is this P681R, which is not only found in the Delta variant, but is also new in the Delta variant. There is this P681H mutation in the Alpha variant, but which is a proline to histidine, but this is a proline to arginine, which is new in this Delta variant, and this seems to confer extra infectivity onto the virus. So now we're going to look at this one, and this is looking at that other mutation I was talking about, which is this leucine 452 to arginine, which is this one right here. So we're looking at just that. And once again, this is one that's new in the Delta variant. It's not found in any of the other variants. And so this up here, B and C, is looking at how tight these three different mutations are binding. So how tight the spike protein binds to the human ACE2. And so with the C here on this figure is the KD, which actually it's tighter if it's lower, which is one of those things in enzyme and receptor kinetics that can be a little confusing. But essentially what the KD is it's the concentration at which half of the ACE2 would be bound with spike. And so if it's a lower concentration of the spike that would inhabit half of the ACE2, then that means it must be binding tighter because you don't have to add as much spike in order to have it be at 50%. And so this L452R is conferring tighter binding onto the spike protein. But then we can look over here at E and F, and this is actually looking at these three different mutations here compared to the parental, which is that OG version, and then the parental, and then parental plus that D614G here. And this is looking at infectivity, so relative infectivity, where 100% is set equal to sort of the parental at highest concentration of the these different mutant versions of the spike protein. And so what we see here is that this L452R drastically increases the infectivity. We can see that this D614G increases it quite a bit too, but then this L452R is actually increasing it quite a bit more. So this is the B.1.429, which is one of the variants that has this L452R. And so we can see that that confers more infectivity onto the coronavirus, onto COVID. And so essentially what I've shown, sort of the takeaway message here, is that these two mutations, this P681R and this L452R, which are both only found in the Delta variant, increase the infectivity of the COVID-19. So probably of more interest to people is actually this study here, which is actually looking at the sensitivity of these SARS-CoV-2 variants to sera from convalescent individuals and vaccinated individuals. So this here in part A is looking at, so these are people who caught the virus before they were vaccinated. So they got sick with COVID-19, then they convalesced, they got better, and then these ones here did not get vaccinated, and these ones here did get vaccinated. And so they actually are testing their blood, their sera, their serum, which is from blood. They're looking at how many antibodies they actually have, neutralizing antibodies. So neutralizing means that it's an antibody that actually works against a particular variant. And so we can see here in the right part where the vaccinated people are, and so M12 meaning that it is 12 months, I believe, month 12 after the onset of symptoms from when they got sick. So it's about a year after they got sick, and these ones in the intervening time got vaccinated. These ones in the intervening time did not get vaccinated. And so what this shows is that there is more of the neutralizing antibody in the vaccinated people than in the unvaccinated people. And that's true for this D614G variant in just that one mutation in sort of the OG virus than the alpha, beta, and delta variants. And so we do see here that there is a reduction in neutralizing antibodies in the blood of people who are vaccinated to the beta and delta variants, but it's still higher than the people who were unvaccinated. And we can actually see on the size here, these concentrations are logarithmic, so it's considerably higher for the vaccinated people than the unvaccinated. These two, B and C, are looking at the particular vaccinations. And so week 3 is week 3 after vaccination, and then W8 is week 8 after vaccination, which is week 5 after the second dose. So essentially what that means is this one here is with just a single dose of the Pfizer vaccine. This is with the full course, the 2 doses of the Pfizer vaccine. And once again, we see that getting the full course of the vaccination is increasing the number of neutralizing antibodies even further than getting just a single dose. And so this one is the AstraZeneca, which I think is just a 1 dose version, but it's looking 10 weeks and 16 weeks after vaccination. And so we can see that there are more antibodies after 16 weeks after vaccination. And so in orange is the Delta variant. And so we can see that these two vaccines are effective against the Delta variant. And then over in A here, we see that just being vaccinated at all is effective against the Delta variant and the other variants as well. And so I think this kind of shows that there's, I think, this sort of sentiment that some people have that, well, I got sick, so I don't need the vaccine because getting sick with it is the same as getting vaccinated. And this is showing that that's not the case, because in both of these here, the people had been sick before, and then these ones did not get vaccinated, these ones did get vaccinated, and the vaccinated person has more neutralizing antibodies than the unvaccinated person, even though in both of these they had been sick with the coronavirus before getting the vaccine in this case. But anyway, I think that was everything I wanted to talk about with this. I hope you found this interesting and useful in your search to better understand COVID-19 and the Delta variant in particular. And I will see you in another video.